Hello and welcome to this Bioprocess International Ask the Expert webcast. I'm your host, Alex Nicolaitis, the webcast coordinator for Bioprocess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we'll begin the Q&A portion, and I'll ask our speakers your questions. Your questions in the Q&A window will only be visible to myself and our speakers. So thank you for joining us today. It's now my pleasure to introduce our speakers, Hagyoung Kim, Senior Scientist, Upstream Process Development, and Dongyoung Park, Senior Scientist, Upstream Process Development from Samsung Biologics.
Hello, everyone, and welcome to today's webinar on innovative approaches to enhancing cell culture density for high-quality productions. I'm Hagyoung Kim, engaging in upstream process development in Samsung Biologics.
Along with Hagyoung, I'm Dongyoung, working at process and platform development of upstream.
I'm excited to share with you the latest advancements in cell culture technology and how our platform, S-Tensify, can be utilized to enhance the efficiency of cell culture processes during upstream development. These are the agendas I would like to share with you today. I would like to first brief you on the biologics market trend and challenges to set the context, then the background of Samsung Biologics' city of business, and finally, introduce our main platform. So our agenda is on upstream process development and cell culture. Normally, within the biopharmaceutical industry, the animal cell culture is adopted to produce biotherapeutics. The city of business is conducted with IND filing in mind as our main objective, where in the short term, we establish a process that makes sufficient productivity for producing high-quality products. The biopharmaceutical industry is currently experiencing rapid growth, driven by the increasing demand for biologics such as monoclonal antibodies, vaccines, and recombinant proteins. However, this growth presents significant challenges, especially in scaling up production while maintaining high product quality and process efficiency. One of the most pressing challenges in cell culture-based production is enhancing cell density to improve yield without compromising the product's quality. And so, we will explore in depth of these challenges, as well as the innovations in scaling up cell culture-based productions. We will also discuss how Samsung Biologics is strengthening the competitiveness of development with our innovative solutions designed to meet the evolving needs of the biopharmaceutical industry. Before going into the technical aspects, let me briefly introduce our comprehensive features of contract development services. As a global contract development and manufacturing service provider, we ensure clients to select the strategy that is best fitting for their development needs through our customized CMC solution platform, which is called Select Tailor. The platform can dynamically accommodate various needs, from cell line development and process optimization to full-scale commercial manufacturing. Our service platform is designed to be flexible and adaptable to meet the unique needs of each client, ensuring efficient development timelines, high-quality production, and robust regulatory support. And even further, through our accumulated expertise in upstream development, we focus on optimizing cell culture conditions to maximize yield and quality, setting the foundations for successful downstream processing and final product formulation. In particular, the upstream process development is predominantly being carried out via the traditional fed batch process, which has contributed to ensuring a 10-month timeline for monoclonal antibody molecules from cell line development to IND filing. So, upstream development is a critical phase in the biopharmaceutical production process. It involves the cultivation of cells under conditions optimized for growth and productivity, which directly impacts the quality and yield of the final product. In this phase, our main goals are to maximize cell density and productivity while ensuring scalability and consistency. Key considerations during upstream development include selecting the right cell line, optimizing media and feeding strategies, and tightly controlling environmental conditions like pH, temperature, and oxygen levels. Addressing these factors early on helps prevent bottlenecks later in the process, ensuring a smooth transition to downstream processing and, finally, commercialization. At Samsung Biologics, we adopt a phase-appropriate quality-by-design approach in process development. In addition, we have in-house analytical capabilities for developing the process across a wide range of different projects. In terms of enhancing the titer, we provide intensified cell culture along with media and additive screening services. We are also capable of modulating critical components of quality, such as molecule glycosylation and charge variants. Traditional fed batch processes are widely used in the biopharmaceutical industry for producing a range of biologics due to their simplicity and well-established protocols. In a fed batch process, cells are cultured in a bioreactor and nutrients are periodically added to sustain cell growth and production. This method is favored for its simplicity and compatibility with existing infrastructure, making it an accessible choice for many manufacturers. However, as we scale up production using traditional fed batch approaches, several limitations become evident. The accumulation of metabolic waste and the depletion of critical nutrients in the bioreactor can create toxic conditions that stress the cells, reducing their viability and productivity. Additionally, these suboptimal conditions can lead to a decline in product quality, which is unacceptable for biopharmaceutical applications. Furthermore, the static nature of fed batch processes means that adjustments to nutrient level and waste management are often reactive rather than proactive. This can result in inconsistent production runs and variability in production quality, posing challenges for large-scale manufacturing. To overcome these challenges, the industry is increasingly turning to more innovative approaches, such as perfusion and intensified fed batch processes. These methods offer a better control over the cell culture environment, allowing for continuous or semi-continuous feeding of nutrients and removal of waste. This not only supports higher cell densities, but also creates a more stable and productive environment for cell culture. In the case of the intensified fed batch approach, it combines the benefits of traditional fed batch processes with the advantages of perfusion systems, and it results in higher productivity and better control over the culture environment without the high cost associated with full perfusion systems. Recognizing the need for more efficient and scalable solutions, Samsung Biologics has developed S-Tensify, our platform that leverages the intensified fed batch approach. S-Tensify is specifically designed to overcome the limitations of traditional methods, optimizing cell culture density for high-quality production. What sets S-Tensify apart is its unique integration of an N-1 perfusion step, which allows us to achieve significantly higher cell density in the N-1 stage before transferring to the main culture. This results in a much higher initial cell density in the main culture, enabling exponential growth and increased productivity from the outset. The core innovation of S-Tensify lies in our use of ATF filters during the N-1 stage. These filters retain cells within the bioreactor while allowing nutrients to pass through and waste products to be removed. This creates an optimal environment for cell growth even at very high cell densities. By applying this N-1 perfusion step, we can increase cell density in the N-1 stage up to 100 million cells per milliliter. This high-density inoculum is then transferred to the main culture at concentrations significantly higher than those possible with traditional fed batch processes. This approach effectively bypasses the exponential phase, maximizing productivity from the start and reducing the overall production time. By applying our platform, the production team achieved a peak viable cell density of 50 to 100 million cells per milliliter in the N-1 stage and inoculated the main culture at 10 to 20 million cells per milliliter. The total integral viable cell density increased by 2 to 3 times, resulting in a productivity gain of over twofold compared to that of the traditional fed batch process. And also importantly, this productivity boost was achieved while maintaining the quality of the antibody produced, which demonstrates that S-Tensify can enhance both efficiency and quality in biopharmaceutical manufacturing. Overall, it is clear that S-Tensify is robust to overcome the traditional fed batch process. Firstly, significant productivity improvements beyond cell line development were evident from the data that we have provided and explained. Second, S-Tensify platform is an excellent solution not only for developers seeking IND approval, but also for those looking to redevelop their overall culture process after receiving IND approval. Even for pipelines in clinical phase 2 or 3, this platform can help optimize the process for enhanced productivity. Since only minor changes will occur, obtaining regulatory approval for process modifications will be easier. And finally, it is an easy-to-apply plug-and-play platform for upstream process development. In conclusion, N-1 perfusion and intensified fed batch have been the key components that constitute the full operation of our platform S-Tensify. Samsung Biologics hopes to support clients in their journey to successfully develop biotherapeutics.
Okay, great. And as a reminder to our audience, you can go ahead and type in your questions for our speakers now. So our first question is, thank you for the presentation on the advantages and data of the S-Tensify platform. If we decide to proceed with the upstream process development using S-Tensify, what would the detailed development process look like?
Well, basically, we go through a round-up optimization run, followed by a verification run. For the optimization run, we start with N-1 perfusion using 2-liter bioreactors, and then we move on to N-process optimization using N-15. By optimizing the N-1 perfusion process, we can expect a reduction in the amount of the media used. Additionally, the culture duration during the N stage may undergo further optimization as well, which can lead to cost reduction. Once the process is optimized, it will be verified again using 2-liter bioreactors.
And our next question is, do you have any additional data on different types of molecules or cell lines beyond what you presented during the presentation? I would like to see more data beyond the examples of case A and case B on the slide regarding the normalized productivity increase.
Case A and case B involve the general monoclonal antibody and complex molecule, respectively. Both cases showed significant increase in productivity while maintaining comparable quality. S-Tensify is expected to tailor the cell culture process to maximize productivity for different cell lines used in biopharmaceutical manufacturing, thereby supporting more efficient, streamlined production. I believe the trend observed with different types of molecules will not differ significantly from the data we presented. As I mentioned, both cases demonstrate a significant increase in productivity while maintaining comparable quality, which suggests we can expect similar results with other types of molecules. For different cell lines, we are still generating the data, However, we will tailor the cell culture process to maximize productivity for each cell line used in manufacturing to support more efficient and streamlined biopharmaceutical production.
All right, thank you. Well, it looks like we are running just a bit short on time, so if you don't get to your questions, we will be passing them along to our speakers who can follow up with you directly. So please go ahead and continue to type in any questions you have, and we'll get those passed along. The last question for the webcast is scaling up possible if S-Tensify is incorporated within Samsung Biologics?
Our company has extensive experience in seamless tech transfer from CDO scale to GMP scale. We also have hands-on experience of M-1 perfusion in GMP scale, which means we have secured the capability to go through late page, such as page 2 or 3. Based on Samsung Biologics, technological expertise and experience, S-Tensify, a groundbreaking intensified batch platform process, is expected to be successfully applied to the GMP scale.
All right, fantastic. Well, thank you so much to our speakers, and thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you will receive a follow-up email providing you with the direct link. We look forward to having you join us at future Bioprocess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.